Welcome to Godlify Foundations, and in this session we'll be looking into osmosis and plant tissues for biology BGCSE. So first of all, I'm going to have my cell here, my plant cell. My plant cell. And in this plant cell, I'm going to put it in a beaker with a solution. So this solution has a higher water potential than the cell. So what's going to happen? Obviously, with osmosis, we're going to see an influx of water. We're going to see an influx of water into the cell from the solution. So water molecules are going to move from the solution into the cell. So what does this, as water moves in, this actually increases volume and pressure in the cell. Then, therefore, the cell now becomes firm. It becomes firm because of now the increased pressure and the volume inside of it. As such, we see now the cell has become turgid. And this is the whole concept of turgid cells. So now let's move on and let's have the same cell. Let's have the same cell. Now this cell, this same plant cell, we are going to put it in a solution. We're going to put the plant cell in a solution. This time, the solution has a lower water potential than the cell. So what's going to happen? We are going to see water molecules moving out of the cell through osmosis. So water moves out of the cell. Because water moves out, this decreases the volume and pressure of the cell, making the cell to be what we refer to as being flaccid. So the cell becomes flaccid, and this is called flaccid cells, where water moves out and the cell becomes more, in a way, weak, but it has less volume and less pressure. So if you leave it for more hours, let's say 48 hours, inside the solution of low water potential, what's going to happen is that the cell membrane is going to detach from the cell wall. And this process is called plasmolysis. So we're actually going to see the cell in a new kind of structure where we have the cell wall here, and then we're going to see the membrane kind of touching, but there are a few parts where it doesn't touch. It is detached from the cell wall. And because it was originally in the solution, we're actually going to see the solution being present in those spaces left after plasmolysis, because the cell wall is fully permeable. So the solution is actually going to pass through into those areas where it's detached. So that's the process of plasmolysis. So when all plant cells have been plasmolyzed, this actually leads to wilting in the plant. That's why we see plants wilting, because of that whole process of plasmolysis. And that was it on regard to osmosis and plant tissues. Thank you.